I want to start by getting into the fundamentals of what flavor is.
Okay.
So can you explain to me on a molecular level what taste and flavor are?
Yeah, well, so flavor is mostly taste and smell. Well, mostly smell, but in addition to taste, and there's a little bit of a contribution from touch and the other senses. But on a molecular level, taste molecules tend to be water soluble and related to like evolutionary drives. So things like sugars, which are an easy source of energy for us, salts, which we need for all of our cells to function, etc. When it comes to smell molecules, chemically speaking, like the one thing they have in common is that they're all like relatively small and they are good at becoming volatile. So like traveling through the air, which is how they get inside of our nasal cavities for us to smell in the first place. But beyond that, there's, maybe a few hundred to a few thousand taste molecules that we can taste. When it comes to smells, I've seen the number estimated at like 40 billion recently, at like the potential number of molecules that we can sense. So there's not really one unifying theme tying all of those together. It's kind of an all-purpose sense for catching like often very tiny amounts of rare and unusual molecules.
So just focusing on taste, we'll say there's hundreds of different molecules that we can taste. There's only 5, 6 different taste sensations, maybe more, don't know. We can talk about that. So how do they sort of split out between the different tastes?
Well, so 5 tastes, sweet, sour, salty, bitter, and umami. Salty pretty much only senses sodium. Like lithium can also actually activate the salty receptor. So for a while, in like the mid-20th century, for low salt diets, for like hypertension, people would sometimes be recommended to use lithium chloride as a salt on their food. That's also a bipolar disorder treatment medication, so that's been discontinued. But apparently it tastes salty. And then with sourness, there's like a bunch of different acids. Yeah, so like lactic, citric, tartaric, malic acid, etc.
Are there any acids that don't taste sour to us?
Well, I mean, there's a lot of molecules that like are have the name acid in their name that may or may not taste sour, just because you're getting sort of into the semantics of like, well, are we talking about a culinary acid or a chemistry acid?
Sure.
Yeah,
Yeah.
but and how acidic an acid, how sour an acid tastes. There's some that are like much stronger than others too. Something like most of the like food acids, so like tartaric, lactic, acetic acid, the organic acids, they taste very sour, but they're actually like technically weak acids. And then things like hydrochloric acid and sulfuric acid are strong acids.
Don't eat those.
No. I mean, they put phosphoric acid into like sodas, but yeah, that's kind of the
but diluted out. Yeah.
Most we play with those inorganic ones. And then with sweetness, there's like many different sugars that we can taste. There's a handful of like what we would call monosaccharides, like very small simple sugars, glucose, fructose, galactose, and then those like combine together to make disaccharides like sucrose and maltose. And then there's some, as like the chain length gets longer, once it gets too big, it stops fitting in the receptor. So there's like an upper limit to.
And is that kind of when you get to starch territory then, or is that something different?
It's kind of like an in-between area. We'd call them oligosaccharides.
Okay.
Instead of a polysaccharide, kind of like oligarchy. It's the same root, so it means like
Okay.
several sugars rather
Okay.
than many sugars.
So they're still carbohydrates, they're just not kind of triggering a taste reception on a sweetness.
Yeah, and then they're not like large enough to do starchy things. And then with umami, pretty much glutamate is the one amino acid. Aspartic acid, I think, it comes in second in terms of like activating that receptor. So there's 20 basic amino acids that like all of our proteins are made out of.
Here's a good example of acids that don't taste sour, right?
Exactly. Yes. So most of them do not taste sour. Some of them taste quite bitter. Phenylalanine I've tasted and it's like disgusting. glycine is pretty sweet, actually, strangely. But
Some of these appear in like protein powders and branched chain amino acids and all that kind of thing. And because BCAAs taste disgusting. So
like leucine and those are not good. But glutamate, which is our favorite amino acid, is like very, very umami. And then I guess aspartic acid is kind of similar in structure to glutamic acid, so you get like a small activation, but it's sort of like a footnote. Then bitterness is where like the sort of menagerie of taste molecules gets like really diverse. Just because bitterness is evolved, I almost said designed, not really designed, but evolved to sense potential toxins. And there's like so many things that could potentially, kill us that we've actually, with most of the other tastes, there's like 1 or 2 versions of the receptor. With bitter, there's around 20.
Yeah.
Because it's like such chemical diversity in the things that can fuck us up. Sorry, can I swear?
No, you can swear for sure. Yeah, especially when it's something as serious as bitterness.
So like, and a lot of them don't have any like chemistry in common. So
is this because plants have basically evolved new ways to fuck us up?
Yeah,
like new deterrents.
Exactly.
So we have to be receptive to it.
It's basically like a chemical arms race. Us and plants and fungi and other other things. And then there's also like chemical processes like burning things that can be quite bitter.
Yeah. Sure. Yeah. Just on so what things are poisonous but not bitter? Like, because that would be the smart route to go down, right?
Yeah, I mean, unfortunately, there's not much of a correlation between like how bitter something tastes and how toxic it is. So yeah, there's a lot of things that are potentially toxic but not bitter.
I mean, I guess everything's toxic in a high enough quantity.
Yeah. But there's like a modified amino acid in some legumes that can like really mess with your cellular metabolism and ultimately kill you, that you like can't taste the difference of. And I think like botulinum toxin doesn't usually taste bitter, so that's like one of the reasons why it's so dangerous,
because you just don't know the food's off.
Yeah, exactly.
Right. What about smell? How do you the smell molecules you mentioned, potentially trillions? Was it?
The most recent academic paper I've read estimated that there are potentially 40 billion smell active molecules, but then because of the like weird sort of computational strangeness of smell, that can generate like trillions of different odors. Exactly, and like different amounts. And there's like synergies and things like that. So yeah, but lots and lots of
let's just go with a lot. And are we equipped to detect those individually? I mean, maybe not put a name to them because that's a lot of words, but can we compute them?
Yeah, I mean, so like your olfactory system is basically like set up to generate a brain signal from like basically anything you throw at it. So it doesn't actually necessarily distinguish between like the signal from a single molecule and the signal from like a mixture of molecules. The way that it's generated is that you have, this like patch of receptors up at the top of your nasal cavity. You have about 400.
That's the epithelium, is it?
The olfactory epithelium, yes, indeed. And there's about 350 to 400 different types of olfactory receptors. And each of them will like bind in different ways to like many different molecules. And if taste is like hitting, one of 5 keys on a piano, you've got like sweet and salty and sour. But just like one at a time. The signal generated from smell is more like a QR code. So you have these sort of like this patchwork of 400 different receptors, each sending a signal. And the signal that gets generated, the olfactory epithelium attaches to the olfactory bulb, and that's sort of the like signal generator for the rest of the brain. It is actually like a 2-dimensional pattern of
because it's a flat surface. It's
and so like all for any of these 400 receptors, all of X type of receptor connect to the same area on the olfactory bulb, so it collects it as kind of a like a spot among like 400 different spots. So some like sensory psychologists would call that an olfactory image, even though we can't literally see it, but it is like technically.
It's almost like a dot matrix printer.
Yeah,
kind of,
yeah. No, and it looks actually quite a bit like a QR code.
Yeah, or a very large chessboard, perhaps.
Exactly,
Yeah,
exactly.
yeah.
Yeah.
So, have we reached the state of the art where we can trigger those receptors individually to bring about aroma sensations in the brain? Are we just waiting on Neuralink to address that particular?
Well, I mean, so we're at a point where we can we've identified the family of genes that make up olfaction. That was actually figured out in like the early 90s, and the two researchers got the Nobel Prize for that because it was like completely unheard of. Like no one had any idea how smell worked. And so once you have those genes like sequenced, you can basically get like a cell in a petri dish to like make a bunch of them, and you can like throw different things at it and poke it and like measure how much of a signal it generates. But in terms of stimulating them, like, I mean, you can throw molecules at them, smell molecules. I don't know that we've necessarily figured out a way to fire them off without using molecules, like a pure, electrical kind of thing. It's definitely the kind of thing like that. There's probably 10 startups working on at this point. I don't know how viable. I mean, like I knew when I was working at MIT, I knew somebody who was with vision trying to develop a way of pinpointing individual like light receptors in the retina to like create a like VR image essentially. But I think we're a long way
Reverse engineer imaging, and that would be a similar thing with smell and taste. Can you speak a little bit about what is actually happening when the molecules touch our taste receptors, and similarly with the olfactory epithelium? Is there a chemical transfer there?
Well, there's a couple of different like types of receptor. For salty and sour, they are essentially like you'd call it an ion channel. It's like a little eclair or a donut where the sodium ions or the acid ions actually like pass through it and then change something inside the cell. For the most part, the other receptors are what are called G protein coupled receptors or GPCRs. And what that is like the outside part of the receptor, the side like facing, the inside of your mouth and the inside of your nose, it is like a specially shaped catcher's mitt. And there are certain molecules it can like grab onto, and once that connection is made, it'll kind of like clamp around it, and that change of shape will then like send a signal through several other bits of the receptor.
Like a Venus flytrap kind of thing.
Exactly.
Yeah.
Yeah.
That's fascinating to think. Every time you stick a bit of food in your mouth, there's this kind of incredible grab process going on.
It's kind of shockingly intimate, actually.
Yeah. It is. It does make you think a little bit more about what you're putting in your mouth when you know that you're kind of... It's almost sexual, isn't it?
Yeah, a little bit. Kinky.
And then from the what's going on with aroma then?
Mm-hmm. Oh, So aroma, I think they've identified are all of these G protein coupled receptors. So there's about like 400 different shapes of protein. So like the outer part of the receptor, they're all proteins. And proteins are like big, long chains of amino acids that can have like stiff parts and floppy parts and like, shaped in very specific ways. So, when a small molecule that one of these receptors can grab onto, actually, like with many small molecules, like let's say eugenol, the clove smell in cloves, there might be like 5 or 10 different types of receptor that it will like interact with, just to like different intensities. So depending on how hard the clamp is, it sends like a weaker or a stronger signal to like the cell that the olfactory receptor is attached to, and then that fires off a signal to like through the kind of the axon, because they are actually like brain cells that are like dangling in your nasal cavity. So that's totally normal. So this like sort of long tail of the cell will then like feed a signal into like one little spot on the olfactory bulb, and then that gathers them all together into a
I mean, again, that is just remarkable to think about, that you've got this kind of pad, which is like, what, the size of a fingernail or smaller?
thereabouts. Yeah.
Yeah. And all of these different shaped arms, I'm going to call them arms.
Yeah.
I'm not a scientist.
Yeah, that's fine. No, I mean, I think like metaphor is powerful.
Yeah, exactly. Hanging down,
Yeah.
waiting for the right shaped molecule, of which there are 10 billion, did we say?
Up to 40 billion.
40 billion?
Yeah.
40 billion different types. And they are evolutionary tuned to grab hold of the right ones. And then it's not just binary, right? There's actually a spectrum of signal that it can then send up into the brain.
Yeah.
Whoa.
Right?
That is insane.
For something that like for 3,000 years we thought was just like an unimportant sense for us.
Yeah. Yeah, like we're not dogs. We just don't need to
Exactly.
be able to do this so much.
Apparently we do.
Yeah. Although when I get a cold, I can't taste much.
Yeah. Well, I mean, anosmia, is the like lack of an ability to smell, which has been like medically considered sort of a curiosity until COVID, when millions of people have been experiencing anosmia, and it's like kind of more of a quality of life issue than other sensory impairments, but I mean, it can lead to like pretty significant clinical depression.
I'm sure. Yeah.
And I mean, we do know from like earlier work in like elderly populations that like losing your sense of smell, or like greatly reducing your smell acuity, can lead to like nutritional issues, because you like lose interest in eating.
Yeah.
It's so boring without our sense of smell.
Yeah, sure. It feeds into our sort of satiety or desire for food so much, doesn't it? And the joy. Mm-hmm. I mean, just kind of going a little bit off tangent, getting maybe a little bit philosophical. Why do you think there's so much pleasure to really derive from food? I mean, obviously, we need to eat to survive. We need sugar, we need protein, salt, and so on. But it feels like the pleasure side of it has been ramped up dramatically. We can get so much joy from that first bite of a burger or a really ripe peach or, whatever. Do you have any thoughts on why it's such a pleasurable thing to do? We'll get back to the show shortly, but first I wanted to give a shout out to our sponsor, Fever-Tree. I have been using Fever-Tree since 2006, when I opened the bar at Jamie Oliver's 15 restaurant in Cornwall, and right through to present day, where we still use Fever-Tree at Blackrock in East London. As a maker of drinks myself, I take very seriously the outsourcing of the sourcing of ingredients. But Fever-Tree are the brand that told us that if 3/4 of your drink is your mixer, you had damn well better pick the best one. Or words to that effect. Right now, it's Fever-Tree Ginger Ale that I'm reaching for. Right now, it's Fever-Tree Ginger Ale that I am reaching for. Right now, it's Fever-Tree Ginger Ale that I am reaching for, which is the perfect Florida whiskey in a whiskey highball. A good ginger ale extends a whiskey out. It dumbs down the alcohol a little bit without muting the flavor of the scotch itself. Fever-Tree know this, which is why they source their gingers from 3 distinct producing countries: India, the Ivory Coast, and Nigeria. Like blending whiskey, each ginger brings something to the table, combining together to offer aromatic intensity, a nice calm spice, and a long depth of finish. To learn more about Fever-Tree, check out the links in the show notes. But now, let's get back to the show.
Yeah, I mean, I think there's like 2 aspects to it. I mean, one, generally, like humans are a very social species, and so like we've evolved to be a social species. So like kind of everything in our evolution you have to look at through a lens of like how does this impact people not being lone wolf but having to have like lots of shared experiences. And smell is like very, very closely tied to emotion. Actually, when we like generate those initial olfactory signals at the olfactory bulb, they get fed directly into our limbic system. Like the limbic system, the like emotional centers of the brain, the amygdala, etc., are actually considered part of the olfactory cortex. So they're so like physically linked that you'll actually have like an emotional association to a flavor before you even consciously perceive what that flavor is. So we're really hardwired to like build emotional associations, both positive and negative, to smells, and smell being the largest part of flavor. So, and a lot of that has to do with like, when you're a child and don't know anything about food and like will literally pick up, poisonous things and poop and put them in your mouth, having a familial happy experience when you're eating something and don't get sick, like you store that like very, very intimately. So like we're wired to do that for sort of like group cohesion and survival, but also a lot of the ability to experience smell while eating. So we call that like retronasal olfaction, because most animals can sniff and smell things in front of their face. For the most part, only mammals have this experience of getting like a variety of smell signals from the inside of their mouth while they're eating, retronasal olfaction. In humans, in human evolution, you really saw an expansion of that like coincided with humans becoming, actually not humans, but like proto-humans becoming omnivorous. So we're not like koalas that only need to eat eucalyptus and only to look for like a couple of signals for our food. We can kind of make a diet out of like almost anything. So
Do you think as we broadened our diet, it became more necessary to tune ourselves to what we were eating?
exactly. Yeah. So we're kind of supposed to eat a variety of things, so we like get a lot of pleasure from activating olfactory receptors with some of these like teeny tiny little molecules that are only like in a peach or something like that. But also like we just have to have the ability to make lots of different things into food and then enjoy eating them.
Just imagine the number of proto-humans that died through natural selection because they didn't have a well-formed enough sense of smell.
Yeah.
To be able to detect when things were potentially poisonous. They couldn't make the correlation between, okay, that aroma there correlates with bitterness, which correlates with poison. Just dropping, like, so many must have died to get to the point where we're at now, where we're kind of pretty good at it, right?
Yeah, no, we're pretty good at it. Interestingly, they think now that sourness, like the taste sensation of sourness, was very similar in that way. That like vitamin C tastes sour, which is great, but also if you consider that like for early hominins, that's what they would call them, pre-humans, most of the food you would be eating would be like contaminated with some kind of microbe, like a yeast or a bacteria. It was a large benefit for you to be able to distinguish between like a lactic acid bacteria, which produces acids to like outcompete other kind of more spoilage related microbes, to distinguish between an acidic fermented contaminated food and a spoiled food. And so these early proto-humans that developed this taste for acid were more likely to go for like lacto-fermented foods over spoiled foods, and then like pass that gene on to their descendants.
Do you think we're good at that now then? You think we're pretty good at detecting the difference between spoiled acid and good acid?
Oh, well, so we just sort of like lucked out that most of the acid-producing bacteria don't also produce toxins.
Okay.
Yeah. Well, either we lucked out or that was the case, and so we learned
evolved
to recognize acid as a signal of like safety.
Yeah. Probably that one, I would think. We evolved to kind of...
I mean, like with evolution, I think it's easy to think of it as like, oh, well, we were like supposed to, or like guided towards, or there was some, I don't know, intention or logicalness, but a lot of it is just sort of like a random soup of coincidences.
Mm-hmm. So 5 taste sensations. I keep on hearing people talking about a potential 6th or 7th, and then there's obviously this distinction between taste and tactile feel and texture and temperature, and then you've got things like astringency.
Yeah.
So can you give us a shape of how you put all of this together and where what box everything sits in, and where the new boxes are?
So we like generally agree that there are 5 basic tastes. Umami being the most recently kind of introduced into that group. So we know that like what the taste receptors are for them. We know like more or less how they send signals to the brain. There are other sensations that are sort of under investigation as like possibly having a taste component.
Sounds like a police interrogation.
we have several suspects of interest.
The arrest warrant's out. Get in here.
Potential taste.
Taste crimes. I know a few people who've committed some taste crimes.
I know, funny, yeah. So it's serious business.
Sorry. So new taste under investigation.
So yeah, some of these suspicious characters include carbonation and fats/fatty acids. There's probably some kind of taste component to those. We're not really sure yet if they have their own receptor or if they like interact with existing taste receptors in some interesting way.
Carbonation. Can we just hold on that one a second, because it's fairly relevant to drinks, obviously, and it's tasty. I was led to believe it was an acid. It's basically carbonic acid that's kind of triggering an acidic response.
Yeah, so that's
sour response.
There is that. So that is one of the theories that are going around. I think I probably should have reviewed the literature before I started talking about this. I think we have like carbon dioxide receptors elsewhere. That's what the sort of like choking reflex is. It's not a lack of oxygen. It's actually the presence of carbon dioxide. So it's definitely possible that these sort of like existing mechanisms for sensing carbon dioxide feed into.
It's interesting because it feels like when we've hacked a little bit, right? To make it enjoyable.
Right. It's a little bit like spiciness, where you say it's supposed to like a signal of danger. That's like cool.
Let's have it.
Let's be masochists. Like a little death every time we drink a sparkling water. So some relationship to acidity, possibly some relationship to like existing methods of sensing carbon dioxide, possibly something else. Still figuring that one out.
Okay, well, we'll keep drinking beer and champagne.
Yeah, it seems to be working out. Okay.
So far, so good. Okay, so carbon dioxide. What else did you mention regarding
fats?
Fats.
Fats and fatty acids. Yeah, they're.
The question with them, it seems like fat feels. I mean, it's often with fat, there feels like there's some umami in there, right? But also there's a textural thing.
Yeah, there's definitely a textural.
So it's kind of pulling that apart on whether or not.
And I think the taste component thing. You look into like deep enough into the literature and you find these studies where like to test smell and taste interactions, they will like seal off someone's tongue and put them in an MRI machine and then like send a tiny tube full of droplets down the back of their throat and then feed them a puff of air at a very specific time. So it gets very like Baroque the way that we pinpoint these things. With fats and fatty acids, I think just through some of these like neurological and psychological experiments, they found that there's like some amount of signal that's like not accounted for by texture and probably comes from taste.
What about metallic?
So so metal ions we can often sense as bitter. So like think like magnesium, calcium, etc. The specific like metallic taste, like if you licked a penny, for example, that's not actually taste at all. That is like purely smell. And you're not actually smelling the metal at all. What happens is that like since metals are like pretty reactive, it will actually catalyze like an oxidation reaction from the oils on your skin. And that will make an aldehyde essentially. Well, an aldehyde, it will literally make aldehydes. Similar to like the smell of blood, that also comes from an aldehyde, that the iron in your blood and in your muscles interacts with like cell membrane lipids and then generates these like funky little smell molecules. So yeah, metallic is a smell.
So if you need to eat a lot of blood or pennies, just hold your nose and it should be okay.
Exactly.
Yeah. Good to know. Let me just write that one down.
Yeah, happy to.
Perfect. I think we're done here. And then, is there any other candidates, any other suspicious characters?
For taste, not that I'm currently aware of, I don't think. Someone will probably write in and correct me, but let's put that out there, correct me.
Yeah,
if
let's go
there
for it.
is one.
Stick it in the comments. What about astringency?
So yeah, astringency is related to touch. So I guess we'd call that a trigeminal sensation. The trigeminal nerve being the like three-rooted nerve that like innervates our face. Interestingly, astringency, sort of like metallic, you're not tasting or interacting with the astringent thing directly. For example, if you like are drinking a very tannic wine and you experience that kind of like leathery feeling of astringency, what's happening is that the tannin molecules, the like anthocyanins in the wine, are binding with some of the proteins in your saliva that help like lubricate it. And what you are feeling is not like directly those proteins or directly those tannins, but like the feeling of your tongue rubbing against the inside of your mouth with no lubrication.
No way. That's incredible. So basically you're kind of WD-40ing all the grease away so that you've got no... Yeah, right.
Yeah.
Okay, wow. That's nuts. Lack of lubrication is what causes astringency.
Yeah. I don't know if you've ever like, you're probably too important to do this for many years, but like emptied out like a spit bucket at a wine tasting.
I'll do my duty.
No, sorry, not that you're above it, but you know, thinking back to your
I've certainly done it.
That kind of like scum on the top. That's precipitated salivary glycoproteins.
Right, so it drops out of the solution and sits on top. So you're right.
It's like
So
a foam,
it's literally people's throat lubrication that you're seeing flowing on top of a spit bucket.
Yep. Charming.
Cool. Well, I haven't emptied one of those for years, and neither will I be emptying one in the future now, or looking at one.
This was one of the. It's like fantastic being a graduate student in a wine program, which is what I was. I did my PhD in the Department of Viticulture and Enology. It also like changes some things permanently for you.
Yeah, actually, we might come back to that in a bit. Actually, I'm interested to hear from you, like when you delve too far into the science, what newer versions come up. But let's come back to it later.
Okay.
Can we focus on sweetness for a second? Because it'd be good to talk a little bit about sweeteners. What can we say about what they are doing and how they work and how they're different to sucrose and fructose and all that kind of thing?
Yeah, I mean, so the sweetness receptor, it's got a protein that is like designed or randomly evolved over millions of years through trial and error to like bind very well to sugar molecules. But I guess like binding to a sugar molecule is not like, it is a process, but it's not like a protein can only do that one thing. A protein has like general functions, like areas of stickiness and shapes and bendability that happen to be very well adapted to sugars, but are sort of naturalistic enough that like other things can happen to be very good at binding to them. So I guess like a corollary would be like spiciness. So we sense spiciness not through taste but through touch, and we have a receptor, TRPA1, that is for basically telling you when you've been burnt. It only gets activated above 109 degrees Fahrenheit. So it feels like painful and hot and burning. And for whatever reason, capsaicin, the spicy molecule in chilies, is like really good at binding to that receptor and firing it off and making it feel like you're being burnt when you're not being burnt. So yeah, I mean, like for any of these things, there's mechanisms that exist in flavor. Either there's like a very specific thing from natural selection and evolution that it's designed to do, or some other random thing that happens to also be good at activating that. So with like artificial sweeteners, a lot of them are just able to either like fit a little bit into that binding pocket. Some of them might activate like a different point on the receptor, but essentially get it to generate sometimes, well, usually like much higher amounts of signal than like a sugar molecule actually.
Yeah, it tends to be that they're packaged in a sort of diluted form to match sugar, doesn't it? But they're actually a hell of a lot sweeter.
So, some of them are like 1 million times sweeter than sucrose.
Yeah, which is crazy.
Yeah.
You just mentioned about chili and heat. Where does alcohol fit into this?
Alcohol I believe alcohol also activates that burning receptor. I mean, that's why it feels quite warm when you're drinking it. There's also like a couple of other like general chemical irritation sensors that it generates. That it activates. For a lot of people, they notice like a bitter sensation, especially if they're like super tasters. So there's like an element of that. And alcohol can actually also be slightly sweet, not super sweet, but like just a little bit. So I mean, alcohol is like a pretty small molecule as like taste and smell molecules go. So it's like, I don't know, maybe not surprising to kind of like work its way into many different places. It's not necessarily like designed to be.
Yeah. Are there molecules that trigger both taste sensations and smell?
Yeah. Excuse me. Some of the flavor molecules in absinthe are famously examples of those. Anethol, which is the like very phenol aroma of fennel and anise, has a very intense smell, and in like high enough concentrations will actually activate the sweet receptor. So like there are many smell molecules that we associate with sweetness, so you'll actually get this sort of like sensory illusion that there is sweetness when there is none. But anethol and some of the other ones will actually activate in high enough
That's
amounts.
interesting, because I was used to kind of, and this is like my sort of pseudoscience, basic bitch kind of nature.
Well, it's okay because nobody's like, nobody, I mean, we've like known this in the sciences. Nobody's told anyone else about it. So like, everyone's had to just kind of make it up.
That's my excuse. Until now, I've not been told by a scientist. But I was used to kind of think, well, if you can't smell it, then it must be a taste. But that's obviously not true. Because tastes, typically you can't smell them, right?
Yeah, so I guess like tastes that you can also smell would be like the exception to the rule. For the vast majority
There's not many of them.
Things are just tastes or just smells. I guess acetic acid would be another example, like because that is very sour as vinegar, but you can definitely smell.
Yeah, so when you smell vinegar, you are smelling the acetic acid.
Smelling the acetic acid.
Okay, interesting. That's interesting. And then are there other organic acids that you can smell as well?
There's actually a lot of organic acids that you can smell, especially like small fatty acids. I don't necessarily know how many of those are actually sour. I think butyric acid might be a little bit sour, and obviously smells like cheese.
Yeah.
And then as you get bigger, they start to smell goaty and then waxy.
That's right. Okay, so as they get bigger, they go from cheesy to goaty to waxy.
Yeah.
Right, waxy. You wouldn't think that would end up there, I don't think.
Right, and a lot of it's like very, what things smell like in smaller versions or smaller concentrations versus like more complex or more concentrated ones is like very random.
Is it?
So there's some, when I was in grad school, I was like a volunteer, experimental subject on some other students' like research projects, including like GC olfactometry, where you use a gas chromatograph to like separate all the smell molecules in something, and then usually you send that through a chemical detector, but you can also like set up a little nose cone on the instrument and have someone like sit and sniff the whole time and tell you what they smell. And there's some components in wine that I believe like hexanoic acid and ethyl hexanoate that will smell like quite fruity and nice in like lower concentrations, but in higher concentrations smell like quite cheesy. Just like and probably what happens is that like there's a couple of receptors that are like very sensitive to them that are always getting fired off when there's small amounts, and then a couple of receptors that are like not as sensitive, so you need like a lot of that molecule to generate a signal. So it only happens when you have
got you.
good.
Yeah. So as well as having 40 billion different aromas, you've also got the potency of the particular aroma
so
that
like
changes.
Concentration dependence. Yeah. And then once you start making mixtures of smell molecules, I mean like mixtures of 3 components, I think they've found in the literature, can have like synergy effects, like literal synergy, not like business talk synergy, although business synergy also exists, but less often than people say it does.
True scientific synergy.
True, literal, statistically significant synergy where you'll actually create smell qualities in the mixture that don't exist in any of the components.
Yeah, so I mean, it seems like we're talking almost infinite number. It's kind of getting in that direction.
I think, yeah, it's like
in all practical sense.
Exactly, it's like one of those very long irrational numbers that like fine, just round it up to 3.
You mentioned about gas chromatograph mass spectrometry.
Yeah,
I think it's the first time I've ever said that without messing up some bit of it. At least
it
I think
was very good.
Right.
Yeah, no, I felt like you talk about this all the time. Could have fooled me.
We've obviously talked about how taste and smell work. So how does a robot smell and taste? What sort of equipment would you be using in a lab to measure this kind of stuff?
Yeah. So to sort of recap how smell works is that you have most smells are a mixture of like a few to like hundreds or thousands of molecules, and they all kind of mix into a soup and bathe your receptors, and they all get activated in different ways, and you generate the signal. That's not very useful if you're trying to like detect what individual molecules are. So what chemistry figured out in like the 50s or 60s was that if you do chromatography, so that's like in elementary school where you took a piece of blotter paper and put a circle of marker ink on it and then put it in a glass of water, and the water would like spread through capillary action and separate the different pigments in the ink from each other. You do that, but in like the gas phase, so a like gaseous stream of smell molecules, and you have instead of a piece of filter paper, it's like a very, very thin tube. We call it like a capillary. It can be like 30 meters long is like the most standard length, but they make like shorter and longer ones. So like very long, coiled into a coil. And it's got this like one of several different sticky substances on the inside, and like different small molecules stick to it to different degrees. So what happens as they are like traveling down this very, very long tube is that they'll start out as a like very jumbled mixture, but then basically in order of stickiness, they'll sort of separate into these distinct bands. And so you've accomplished part one of the task of analyzing these, which is like separating them from each other in the first place. Then you have to try to get an idea of like what they actually are. And
sorry, just to recap that, they stick based on their weight or their concentration along the tube?
It depends on. Sorry, I'm getting like, well, it depends on like very many surface area and polarity and.
Sure.
No, they based on their chemical characteristics.
Okay.
Yeah, and so so it's like concentration independent. You'll just get like a bigger band.
Okay.
If there's more of it. Yeah, and so then we actually like the way that we detect or sort of identify what these things are is kind of like weirdly indirect. I think like a lot of things in science are actually this way if you look deep enough, just like most people don't have the opportunity to. So we think like, oh, you just press a button on the CSI machine and it says like, it's definitely this molecule. So if you've ever wanted to know how a mass spectrometer actually works, is that you have this, nice separated sequence of bands of molecules, and you push them out into this chamber and then fire like a beam of electrons at them, like very high energy electrons, which smashes them into pieces. And then the machine sort of like counts how many of each size of piece you have. So you get, so that's the mass spectrum.
Yeah.
Exactly. Mass spectrometry. And so then what you have to do if you're doing GCMS is like look into a database of basically people who have been nice enough to do mass spectrometry on every molecule they can find and kind of generate like it looks kind of like a barcode. So it's like a list of like barcodes, fingerprints, and then you take the signal that you've generated and then compare it to this whole database and then come up with the potential matches for it.
I mean, you would think it's a hard thing to do to analyze smell molecules in a food or a liquid or whatever, but the way you described it, it sounds even more complicated than I thought. Like there's a lot of work there to get
a lot of work. And like chemistry is sort of charmingly old-fashioned in some ways. Sometimes it's not charming, which is that like there's definitely like it shouldn't be this hard Well, I mean, like the mechanisms of it are the way that they are. Like, but
Is this a cry for help?
Shouldn't
be the time.
Yeah, the computers and the software have not caught up to where like biology is.
Yeah.
Just because like it's kind of like an esoteric and expensive thing to want to know like what all the smell molecules in something are traditionally. Anyways, but yeah, no, I as a graduate student spent a lot of time in front of a computer, like zooming in on like, well, we call them peaks because they're sort of shaped like a peak, like individual peaks on this chromatogram, of which there were like hundreds, and trying to get a better identification on.
How long does the process take, typically?
Just to like do the instrumental part.
Yeah, I mean, I've got a sample. I give it
Yeah,
to
you got like a sample of wine. Depending on so you have to like, the step that I didn't talk about is first you've got to like separate the smell molecules from all the other molecules. You can't like put like tannins or fats or proteins inside of a gas chromatograph because it'll like burn it up and
right.
So in the 90s, so you used to have to do like a solvent extraction. So you'd get like a bunch of hexane and like shake the wine with the hexane and then concentrate it down, and it was like very messy and took a long time. In the 90s, they invented this method of taking like a very thin fiber attached to a needle, and you'd get like almost like a sealed bottle of wine, but like a very small vial. And you'd coat the fiber in like chemically sticky stuff. And so you could just, instead of like doing all this crazy stuff with like rotary evaporators and hexane and other solvents, just like stick this little needle in the vial and all of the smell molecules stick to it. And then you can like pull it up and then put it in the gas chromatograph. So that takes like, half an hour, but like there's like a robot on top of the instrument that does it. And then
right, it's not literally just like you're kind of very carefully hanging this thread.
You can if you're unlucky, if your lab did not buy an autosampler. But for the most part, yeah, we use that. And so then like, I feel like when I was doing it, most of my GCMS programs ran like 45 minutes.
Okay.
Depends on how much stuff, how complicated the sample is.
Yeah. And are there any other tools that are kind of common in labs like this which you might use? And as are things progressing? Are there now better GC mass spec machines?
Yeah, I mean, well, if you want to get into like, are you talking about like a single quadrupole mass spectrometer or like a triple quadrupole for like more targeted analysis? There's like time of flight and Orbitrap mass spectrometers. Basically, just different strategies for either like breaking up or counting the bits.
Right. Okay.
I guess a whole different method for analyzing volatiles would be like electronic noses, which try to basically mimic the way the nose works. So you have, instead of separating a mixture, you have like lots and lots of different sensors that you then throw a smell at and then try to figure out like what's going on based on the mixture of signals it generates. But it's like... That's better for like if you know that you're looking for something in particular,
right?
Then it's like a pregnancy test kind of.
Yeah, got you.
It can't really tell you like of a mystery sample what is everything that is in it.
Got you. It's more like a positive or negative than a.
Or like yes, we've seen this before. This is what it.
Okay.
This is what it is of these 30 things that we know. I mean, there's actually some like really interesting work going on in the realm of electronic noses. I know somebody who was actually like. The sensors that he was using, he found a way to get like the proteins of olfactory receptors like to basically live on a chip.
Wow.
And send a signal that way.
This is what we're going to need in like humanoid robots when they start cooking things for us.
Yeah, because how are you going to smell? I mean, because like a lot of things we can smell like at parts per billion or lower.
Yeah.
It's actually been quite a problem that until like quite recently, there were many things that were almost impossible to detect on a GCMS. But we could smell because we were actually much more sensitive than the instrument.
Incredible.
Was. So like wine, cork taint in wine is one that only recently.
It's only recently started to pick it up.
Well, like reliably.
Yeah.
So if you had like a lot of it, you could see it, or you had to do a lot of like crazy stuff to concentrate it to be able to find it. Yeah.
Well, it'd be good having a robot sommelier that could detect cork taint before pouring me my...
It would be. Unfortunately, yeah, trichloroanisole, the smell of cork taint, is one of these things where it's like actually more like parts per trillion.
Is it? That's lower, is it?
Yeah, even lower than that, yeah.
Pesky cork trees, that is incredible.
Yeah.
What you mentioned there about I was going to do this more at the end, but actually since we touched on it, sort of biotech and AI, what do you see in the next kind of 10 years, if anything, are going to be advancements in the way that machines and biology and chemistry can kind of combine together to, for whatever purpose, whether it's for, food safety or to generate new taste or aroma molecules, novel ones. What is it looking like at the moment?
Well, it's funny because you asked me that question like 4 or 5 years ago, I'd be like very pessimistic, largely because there's been like, obviously smell is interesting. Smell is like a cool thing. So there's been a lot of people who don't specialize in smell, but do specialize in some kind of, like machine learning or like network analysis that are like, cool, we can figure this out just by throwing a model at it. And a lot of, this like AI, machine learning, data analysis stuff, it's garbage in, garbage out. So like if you don't have a good data set that you're putting into it, you're not going to get like good answers out of it. And like smell molecules and smells and volatiles are like so slow and costly to analyze that for the most part there just has not been good enough and comprehensive enough data to get anything useful out of this. Recently though, there's been a couple of really interesting research papers where the Monell Chemical Senses Center in Philadelphia, which is like one of probably the world's premier research centers on smell and taste, have collaborated with like AI researchers and chemists to come up with like very large, very comprehensive data sets on like the smell of individual molecules. So you have to actually like train people to describe what they're sensing, which if gas chromatography sounded like a slow process, this is like an order of magnitude slower. It's so slow. So a lot of people don't do it because it's like, why would I spend months to get like 5 data points? But yeah, no, as of a couple years ago, there's been finally some like very comprehensive research on the back end to feed good data into these models. So there's a startup now called Osmo that spun out of this that is doing some really interesting research on like predicting what sort of unknown molecules will smell like. They actually like have the data and the like computational stuff to back it up. So I was did I sign an NDA? I've smelled some of their stuff and it's cool. They're actually like working with perfumers to be
Great.
Let's figure out, and it's sort of like screening for like novel drugs, essentially.
Yeah.
You'll have like
sure.
People in the pharmaceutical industry have been doing this for like years and years.
So they're kind of running simulations of what it will.
Yeah, trying to predict like what. Sometimes it'll like some of these models will look at, okay, how will this molecule interact with like receptors that we know about based on how other molecules interact.
Other similar shaped molecules, yeah.
Sometimes they'll take a much more like bird's eye view of like, well, let's not look at the mechanism specifically, but just by analyzing data on thousands of molecules, like what does this probably correlate with? So there's a couple of different approaches.
It's basically computer aided trial and error, right?
Yeah. It's like protein folding or some of the like the SETI at home program. We used to have it when I was a kid as like a screensaver. You'd use like SETI would the search for extraterrestrial intelligence.
Yes.
All of this like radio wave data that they'd need to crunch numbers on, and so they'd send little packets to your home computer so you could use up your downtime to help them out. So kind of like that.
There was a study published 5-6 days ago.
Okay.
I don't know if you've read it, from Penn State.
Okay.
I'll read you the extract. So the researchers provided a neural network with 20 specific parameters to assess, all of which are related to how a sample liquid interacts with the sensor's electrical properties. based on these researcher-specified parameters, the AI could accurately detect samples, including watered-down milks, different types of sodas, blends of coffee, and multiple fruit juices at several levels of freshness, and report on their content with greater than 80% accuracy in about a minute. Then, this is what one of the authors of the paper wrote, was quoted saying, "After achieving a reasonable accuracy with human-selected parameters, we decided to let the neural network define its own figures of merit by providing it with the raw sensor data. We found that the neural network reached a near ideal inference accuracy of more than 95% when utilizing the machine-derived figures of merit rather than the ones provided by humans." So they basically like let it have at it.
Yeah.
And it was better, or nearly perfect. And obviously this study was based around food safety or food fraud.
Yeah.
So like diluting products out or products that have gone off. But you can kind of see how you could use the same technology to then create, new combinations of flavor, or possibly even identify the flavors or aromas of like novel molecules.
Yeah. I'd be really interested to read that if you can send that to me. It sounds so like there have been kind of like the electronic nose type investigations in chemistry generally for like rapid analysis, like ultraviolet visible spectrophotometry. So basically not mass spectrometry, which is its own like specific thing, but basically like shooting different types of ultraviolet and other light at a sample and seeing what bounces back. There have been, you can actually buy, if you're like a winemaker, like a Foss wine scan. It will, you'll take up a sample, it'll shoot like a bunch of beams of light at it and then get this very complex signal back. And what they've done is actually like collected like comprehensive samples on like many different types of wine, and then done the full chemical analysis so that they can then like correlate the complex signal to what they've already measured. So like that kind of approach in terms of like taking a complicated signal and then trying to, predict what it is based on a database has been happening for a while with like spectroscopy data, but
done manually, like
done manually.
Or looking for simple patterns rather than.
What I'm interested in is like what their sensors are, because that sounds like an interesting thing. I mean, part of the problem with not the problem, like so spectroscopy, it's like it's good at detecting whether like is this sample of like baby formula, does it actually contain protein or does it contain melamine instead? Generally, like flavor molecules are like so such like tiny amounts that like any signal that you get from them is drowned out by everything else. But if they have like different and unusual types of electronics, there's definitely some weird and interesting stuff they could be doing with that. But then also like with some of this like detection of adulteration, like with machine learning models or like neural networks generally, sometimes it will like detect what we would notice about it. Sometimes it will detect something like completely different that just happens to always be correlated to the thing that we're interested in. So so that's with the like non-scientist defined inputs. It just like lets it figure out what is always, different among these things, which is sometimes, especially like for, sometimes like the molecule that they're interested in, sometimes it's just like some random thing that always goes along with the molecule.
Yeah, it feels like the most useful outcomes with this are going to come from combining data from like GC mass spec along with human sense data, like quantitative human sense data, to recognize whether or not it's actually relevant to us.
Well, I mean, the thing that they'd always teach us in flavor chemistry is that like you don't actually know about anything about flavor until you feed it to a human. So, I mean, once we I don't know, gather very, very comprehensive data about like what every possible mixture of little things actually tastes like, then we can start doing some like really interesting predictive stuff. But that's like not a lot of funding for. Maybe now there is, now that there's AI attached to it. But
exactly. You got the machines to do it. I want to move on and talk a little bit about how flavor's made.
Okay.
So I figure we've got different classifications of aroma molecules, like phenols, and then you've got different methods of getting there, and we've got things like Browning reactions, like Maillard reactions. I definitely want to talk to you about fermentation as well. So where do you think the best place to start is here with how flavor is made?
Wow. I mean, we can actually start with like herbs.
Okay.
If you want.
That's a nice place to
yeah,
start.
herbs and
Yeah, that
spices.
sounds lovely.
So yeah, a lot of the tastes that we like from herbs and spices, like thyme or clove or rosemary, comes from like two different families of molecules. One of them is called terpenes. One of them is called phenolpropenes. It used to be that no one knew what I was talking about when I said both of those. People still don't know what phenolpropenes are, but everyone knows what terpenes are because of cannabis and fun stuff like that. But yeah, two different types of smell molecules, and I guess it's easy to think about like nature as a machine that like does specific tasks. So a clove plant is going to like basically have a system specifically dedicated to generating clove aroma, which is like not really how it happens. What happens is that there's various metabolic reactions that all cells need to do to live. So you need to like generate energy from sugars, and you need to generate proteins from DNA and RNA, etc. And there's various like intermediate steps to get there. And so what'll happen is that like just because the inside of a cell, whether it's a human cell or a plant cell, is like essentially just like chemical soup. Everything's like mixed in there together. If you have like a bunch of one of these random intermediate precursors, it'll like bump into an unrelated enzyme and make a random new molecule. So with smells like clove and cinnamon, these come from phenolpropenes, which is a sort of like an area of compounds that also includes lignin, which is like the woody part of wood. So all of those come from the amino acid phenylalanine, so it's called phenolpropenes. So like basically the cell is making a bunch of this amino acid anyway, and other parts of it sort of like siphon off that and then do different chemistry things to it, and you'll generate like a bunch of different small volatile molecules, which will make like clove smell, vanilla smell, etc. And then other parts of it will then like smash those together into like very large networks, and then from the same precursors you get like wood.
So are you saying that those aromas are made in the plant by chance, by accident?
Basically they like evolved by accident. So like phenylalanine or like one of the things leading up to it. So like, sorry, let me backtrack. The way that we teach students, I guess, about how like enzymes and biochemistry work is that like, the enzyme has an active site that is like designed specifically to bind to one thing and then catalyze a specific reaction. And that does happen, but a protein is a blob held together by like different types of sticky bits, and so it can do one thing very well, but it's not actually designed to do that thing. It just happens to do that well. So often by pure chance or through like a simple mutation, it'll happen to be able to interact with something totally different and then make a byproduct. Often that is like very bad for the cell and it dies, but like occasionally it's very good for it. So like it will accidentally produce a tiny molecule that is volatile and acts as like an insect repellent, and then that plant has a better chance of surviving, and then passes on that weird mutation to its descendants, and then you have totally from this random occurrence, this ability to make like clove smell from like an amino acid that every plant
So you're saying it initially happens by accident, but then
and then it is like selected
then
for.
it's like, okay, cool, this is actually pretty good, guys. I've just like survived long enough to
Yeah.
reproduce, so let's make sure the offspring can also do this.
But that's basically how all evolution happens. Yeah,
sure.
but
So I guess what I was just getting at is like, it happens by accident initially, but it's kept because the gene's selected to keep it.
Yeah.
Because it has a reproductive or survival advantage.
Exactly. And then you get this knock-on effect of like, okay, we have this ability now, and then now that you've like created a new stream of molecules, then those might get altered by later mutations, and then you get this whole ability to generate like hundreds of aromas from like one little amino acid.
And so something like clove oil or cinnamon, has it been kept because it is an insect repellent or something along those lines?
Yeah, so most of the flavors from herbs and spices that we like a lot are insect repellents, sunscreens, antifungals, basically like chemical weapons in a way to the plant. We just, evolved millions of years after all of these volatile molecules were already around, and like in a kind of like dose defines the poison way, like it's possible if you eat too much nutmeg, famously you can like get hallucinations and then poison your liver.
You ever done it?
I have not personally, but it was like there was there was always that like one group of kids in high school that was like, yeah, I was gonna try nutmeg. And
I don't think you even need that much, right? Like a whole nutmeg, I think is enough to have a not get liver poisoning perhaps, but you can feel it.
To get
Which
that's what I've heard.
Pretty crazy. Like you can go buy a jar of like 5 nutmegs from the store for a
Right.
couple of pounds.
I think it is harder to eat a whole nutmeg than
Yeah, you got to grate
you
it.
think. Well, you've got to grate it, but then consider that we'll use like a quarter teaspoon of nutmeg
Sure.
for like an entire dish.
Oh yeah, you by accident you're gonna have to go for it.
So if you want to eat like 2 ounces of nutmeg, that's like a very intense experience.
It's gonna be a very nutmeggy.
It like lets you really feel what it's like to be an insect trying to eat a bunch of nutmeg.
You'll get an idea of like what the
You're like,
nutmeg was trying to achieve.
This is a repellent.
Yeah. Unless you've unfortunately evolved to be a human that really likes the flavor of nutmeg,
Right.
and then obviously natural selection is going to take you out, isn't it?
Yeah. Well, and then some of the small molecules in like citrus skins, if you get too much on your hands or on your skin, it'll actually create like phototoxicity, so you'll get like weird intense sunburns from them. So in small amounts they're fun and delicious. In larger amounts, we are not immune to their chemical weaponry.
That's crazy. Don't use orange zest as sunblock. Is like
You'll often see it like when people go on vacation and make a lot of margaritas. They'll get like lime oil and lime juice all over their hands, and then like they're wearing a bathing suit and they touch their skin and they'll get this like weird shaped sunburn from that.
You've been drinking margaritas. Yeah. Also the fact they're staggering around the place and grinning.
Right.
Yeah.
Don't forget to put on sunscreen.
I didn't put sunscreen on basically. Duck.
And you've got a handprint shaped for
I will keep an eye out for it though. That's so interesting. So herbs and spices. So they're pest repellents.
A lot of them. Yeah.
A lot of them. Yeah.
Yeah.
Which is fascinating, isn't it? Because I guess because we're larger than insects, we've got a tolerance for it in small quantities, and yet we've exploited it to so much. They make up such a huge part of cuisine wherever you are, really.
Yeah.
Apart from maybe Ireland, where you're going next. I'm joking. Sorry, I'm half Irish, so I can say this.
Pretty sure they have like parsley.
Yeah. No, they do. I'm being mean. Yeah. What else have we got then besides herbs and spices?
So herbs and spices, make stuff that way. There's a lot of examples of like in living creatures, whether they're like plants or fungi or microbes, of I have this enzyme, let me just like alter this molecule that's around and it'll make something that smells amazing, just sort of initially by accident and then eventually selected for. Then there's a lot of like chemical reactions that make smells. So actually going back to like lignin on wood, You know, if you're burning a piece of wood and you notice like vanilla and clove notes to the smoke, that's actually because when you like break these large molecules apart, some of the internal components are like the same molecules that are in vanilla
Yeah.
and cloves. So you're actually generating like
Vanillin and
vanillin and eugenol.
Yeah.
Yeah.
Yeah, and that's relevant to barrel-aged spirits as well, right? And wine. Yeah, because it's the spirit is doing kind of the same thing. It's just kind of extracting it out and breaking it down. Is that how it works?
Yeah, well, so for most barrel aging, well, you have to use fire to like bend the barrel, so you heat it up.
Yeah.
So just incidentally, from the heat of bending, you'll generate smell molecules, and then most barrels are like intentionally toasted.
Yeah.
Or like in the case of like bourbon, often like burnt
Yeah.
on the inside.
So it's only extracting from the toasted component, not from the wood itself, like as you go deeper into the wood.
Yeah. For the most part, I don't think alcohol is chemically powerful enough to like break that stuff
Got you.
down, but
Yeah.
it will just. So I mean, you're kind of making a slow infusion of toasty wood
Yeah.
into liquor.
Why do you think animals haven't evolved to be less tasty?
Well, so that's okay. So
They could easily have like toxins in their flesh, right? I mean, not easily maybe, because then they've got toxins in their flesh. But
Well, like I guess imagine you're not an animal, but you're like a pepper plant, and making anything besides energy from molecules, like it takes up resources, so there's got to be a good reason to do it. So you've spent a lot of time evolving this ability to make like capsaicin, this like really spicy molecule, and it fucks up anything that tries to eat you, and it's great. And then humans come along and well, actually, you've evolved it so like birds can't taste it. So birds will like delicately spread your seeds. Nothing else will eat you because it hurts.
Really? So they're basically evolved for birds,
Yeah,
but nothing else.
Exactly. Yeah. So birds can't taste capsaicin. So you often see like bird seed coated in pepper extract so that squirrels can't eat it, but birds will.
So sorry, just to linger on that, and I know you're not an evolutionary biologist
dabble,
exactly, but do you think that's just because they want a broad dispersion of their seeds, the chilies, or do you think because you could have mammals doing exactly the
I
same?
think also birds don't chew very much, so
Right.
it doesn't like
They just hock it down and then
it doesn't break up the seeds.
seeds, it comes out.
So it's like benefit on many levels.
Got you. Okay, sorry. Continue.
No, it's okay. So you've been this way for like millions of years, and you're quite happy. And then humans come along, and the capsaicin does work on them. It does burn and hurt, but like they're all masochists, and their brains will release endorphins when they get hurt. So they find that when they eat your fruits, like it hurts, but it feels amazing. So this like very specific and intricate chemical machinery you've evolved over millions and millions and millions of years to protect yourself now becomes like the reason you are getting eaten. So I guess
Sucks to be a chili. Yeah.
well, I guess it does, but now also there are more chilies grown
We do
now
grow them,
than would ever
domesticated
grow wild. So it's sort of
It's worked out okay.
positive, negative.
There's a good
mostly
pivot
positive.
there on
Exactly.
on the part of the chili, yeah?
It worked out okay. So I mean, maybe over like 10 million years, an animal would evolve something that is toxic. I mean, there's like frogs that have that ability.
Yeah.
But I guess considering that most like animals that we use for food, they're not actually subject to natural selection. They're subject to artificial selection that like we are doing. That's unlikely to.
Yeah, and also unless you're breeding them, then there's no way of them breeding.
That's what I mean. Yeah. Is that if a cow spontaneously developed an ability to poison you, like you wouldn't breed other
breed
cows
that
from
cow,
it. You'd be
yeah,
like,
yeah.
what the hell?
Fucking
Taking that one out of the breeding queue.
get out. Yeah, exactly. I suppose, yeah. Cows that can jump over gates with the... What about Maillard reactions? Because it seems like that is something it's a pretty big deal in our sort of food culture, is this sort of cooking, browning reactions. We've obviously evolved to find them highly desirable. First of all, I'd love your thoughts on sort of the evolutionary part of that, and then maybe let's get into actually what's happening. Why do you think it just basically came as a result of man finding fire, starting to cook our food, developing bigger brains, smaller digestive systems, cook food, is good?
Yes. No, I think there's some very compelling arguments from anthropology and archaeology that humans, or probably even like proto-humans, like had to develop cooking before we could evolve into humans for, basically to get enough energy out of our food without, there's some like interesting, I read one paper where they were looking at different like extinct ape skulls and proto-humans and like measuring their teeth, and developed an equation to predict how many hours a day they would have to spend chewing based on tooth size.
Chew cycles, yeah.
And for the early humans, the number it came out with was 28 hours a day. So like that's clearly impossible. So they had to be doing something else besides just chewing raw stuff.
Yeah.
And the answer is cooking.
Yeah, and I've heard this one in it flies around the sort of vegan argument as well, where you just don't really have enough hours in the day to complete the necessary chew cycles to get the nutrition you need out of plant foods. This is not today, but going back, millennia.
Right.
And so meat had to be a part of the diet for them to survive. Otherwise, just isn't enough time because it's calorie dense.
Right. Yeah, I find, Like, unless you had like a lot of avocados available, I think.
Shitloads of like bananas and avocados and
Yeah, but even then, when not even early humans, like anatomically modern humans, we'd eat the whole animal. Like often, like the brain would be of like a wild animal, so not one that's like bred to be, fattened, would be like the most energy dense part of the animal because it's basically like a blob of bacon, it's like mostly fat.
That's a good way of selling brain meat.
Yeah,
blob
exactly.
of bacon.
So
I've never heard it put that way before. It's not the first thing that springs to mind.
Electrified hallucinatory bacon.
I've never eaten brain. Have you?
Yeah. Like cow's brain and stuff. I guess it's it's like it has no muscle, so it doesn't have like a texture like bacon, but it is like a very soft kind of like gel.
And can it be cooked in such a way to give it a crispy edge?
Often you'll like bread it and then
okay. So
like pan fry it.
Yeah, that sounds good.
It is good. Yeah.
Yeah, it's like a brain croquette.
Yeah. Probably not good to eat too much of it just because of like prion diseases potential, but as an occasional treat.
Yeah, as a treat.
It's like a French thing.
Yeah, of course, yeah. I think I've eaten most organs, but or most commonly eaten organs.
Yeah.
But never had brain, never been offered it.
I'm sorry.
I'm just drawn to like Indiana Jones and the Temple of Doom, the monkey brains.
Right.
And there was a food, you might have come across them. Have you come across Bumpus and Parr?
Oh yeah. I've never met them, but I know their work. Yeah.
They've been doing it in London for a while. They do kind of like food art installations.
Architectural jellies.
Yeah,
and things
exactly.
Like that.
And I remember, I didn't go to it, but they did a special dinner where you watched Indiana Jones and the Temple of Doom, and you ate all the food that they get fed.
Amazing.
So the monkey brains, the snakes, like pouring out the snakes. There was eyeball soup, and then of course he ends up drinking blood later in the film as well. There might be something else. Oh. They get fed food at the little village at the start as well. Anyway, that sounded like my kind of thing. It's like
it sounds amazing.
Yeah, experiential food.
Yeah.
Anyway, we were talking about cooking.
Talking about cooking. Yeah.
We got onto brain. So, calorie dense brains. So, what is going on when we cook food?
Well, so when we cook food, you're essentially blasting the chemistry of whatever ingredient you're cooking with heat, and heat when molecules absorb heat, they basically start moving faster. They'll like vibrate a bit more and move through space, and as they're moving through space, they'll like crash into each other. And so the more heat you add, the more energy they'll be doing this with, and so like eventually you'll start getting crashes between molecules where they'll stick together and then make a new molecule, or like you've added enough heat to a molecule that it's vibrating so hard it'll actually like break apart into smaller molecules. So like if you start making a caramel and you're just heating up like just pure sucrose. The kind of like initial like sweet shop cotton candy smells you're producing are actually like most of a sugar backbone with like a couple of bits kind of lopped off from the heat. And then as you add more heat, they'll break down more, they'll like rearrange, they'll combine with other molecules and make kind of these more complex, ultimately bitter smelling and tasting things.
Yeah, they get to bitter eventually. So that just a continuous breakdown of the molecule until it forms something tasting bitter? Or how does that work?
It's like breakdown and then like building back up from those breakdown products.
They form into new...
Yeah, so especially like the pigments, like the colors, the like browns and blacks, and the bitter tastes come from these like larger molecules that take longer to synthesize.
And caramelization, does that fall under the bracket of Maillard reactions, or is that different?
So the Maillard reaction would specifically be related to sugars reacting with amino acids.
Okay.
So that's like the key initial step. So a lot of the like smell products are in the Venn diagram between caramelization and Maillard reaction. There are a lot of molecules in common just because like you have sugars in the mix. But amino acids, well, they all have nitrogen in them. That's what makes them an amino acid. And some of them have sulfur also. So like cysteine, methionine, those are two sulfur-containing amino acids. So you'll make these molecules that are shaped kind of like caramel-related ones, but have like a couple of nitrogens, a sulfur, and then have kind of either like nuttier, more like roasted as opposed to caramel aromas, or a lot of the sulfur compounds actually have these very like intense like roasted coffee, roast beef notes to them. So yeah, it's different chemistry, slightly different smells.
And like, it's worth sort of emphasizing that basically everything tasty, or not everything tasty, but a lot of tasty things, this is going on, right?
Oh yeah.
Coffee and...
Anything that like turns toasted or brown is going to have most of its flavors from the Maillard reaction.
And when you start to think about what food you like, you're like, oh yeah, that's kind of nearly everything, apart from maybe like fresh vegetables.
Right, exactly. Coffee, chocolate, toast, well, bread generally, like roast chicken, roast beef.
Popcorn,
yeah,
like toasted nuts.
Brown butter.
Stop that.
Sorry.
Mic drop, brown butter. So yeah,
Potato, or like roasted potatoes.
Yeah. Oh, all right, now I'm getting hungry. Yeah.
A hazard.
What about smokiness? Kind of extending on from the cooked thing. It feels like that should be undesirable. Is this another sort of masochistic thing, do you think? Or and also like in some foods it's considered to be a bit of an off thing, whereas other times it's...
Oh, like phenols?
Yeah.
Yeah, I mean, so smokiness, so a hypothesis is that, animals before humans could sense the smell of smoke because it was like important to be able to get away from wildfires. And that when humans figured out how to, or proto-humans figured out how to like harness fire and use it to cook food, they had this like existing ability to sense smoke, but then started using it as like a positive association because it was like, this is what cooked food smells like. That's the smell of cooking, so that's good. So yeah, I mean, you only need like very tiny amounts of the main like smoky molecule. There's many, but like the quintessential one is called guaiacol, and you need like a very, very tiny amount of it to get a smoky flavor. But then it is one of these things that once you reach like a high enough concentration, one, if you've smoked something too much, you can actually like start introducing like tars and carcinogens and not so nice stuff to it. So there's like a sweet spot where it's before that,
not dangerous,
where it's good before it becomes dangerous. And you'll also see the transition from like pleasantly smoky to like licking an ashtray.
Yeah. I've often wondered if the sort of attraction to smoke comes from the many generations that cooked over fire, and where all food will inevitably be imparted with a smoky quality. And then maybe like when we taste smokiness, it's a bit like when you sit around a campfire, and most people don't do that very often. And when you do, you sort of stare into the flames and you're with friends, and there's something very kind of peaceful about it and very sort of meaningful.
Yeah.
And I think probably because it's, we've got thousands of generations of ancestors who did exactly that. And there's maybe some programming there.
Oh, I think there's got to be.
Yeah.
I mean, I figure like a fire was, it was warmth, it was food and nutrition. It was also like safety. If you're sitting around the fire, it scared larger animals away. So there's the like survival, nutrition and food, and also this like communal social aspect to
it's
it.
kind of all the human values there in one, isn't it? Yeah.
Everything you need.
And smoke probably comes along for the ride, I should think. It's certainly popular at the moment. Or I don't know, maybe it's waning in popularity. What do you think? It feels like we had a massive kind of barbecue period there about like 5 or 10 years ago.
Yeah, I mean, I don't know that barbecue will ever go out of
True. Well, I mean, barbecue, of course, evolved from exactly what we're just talking about, right? Yeah. But there were a lot of barbecue joints sounding out.
But,
Yeah.
I mean, and like, I know for a while, even earlier than that, like, oh, let's, you use a smoking gun to put just like a little bit of smoke on this thing. And I mean, I think it's like one of these fads where like, it is not delicious in all applications, just takes a long time to figure out what they are.
Yeah.
So like, if it wanes in popularity, maybe we're just figuring out where it really makes sense.
Yeah, sure. That's like a lot of these things, isn't it? You try stuff out. Someone tries it with everything, and it gives you the chance to go, yeah, that actually sucks. Don't do that anymore.
Yeah. I mean, like fermentation is.
Fermentation.
Yeah.
Let's talk about fermentation. I was going to go there next anyway, and you did it for me. I mean, this is kind of, one of your specialist fields, perhaps?
Yeah.
So, and again,
done a bit.
it's one of these things that kind of crops up in much more stuff than you think, right? Like, it's kind of everywhere in our food environment. People don't think about bread as being fermented often, but it's like...
Well, and going back to like the Maillard reaction, people don't think about chocolate or coffee as being fermented, but it's like absolutely essential for like their flavor to go through the fermentation stage.
So what's going on here?
What's going on here?
In fermentation?
Well, biochemically, microbes are making their home in different ingredients and eating up usually sugars, sometimes other stuff, and then converting it into, waste products, but also delicious byproducts.
I guess most people think about yeast as one of these microbes, but what else are we talking about here?
I mean, the really big one is lactic acid bacteria, and there's like hundreds, at least, of species of lactic acid bacteria, specialized to different like niches, but bacteria that eat up sugar and other carbon sources and make acid out of it are, as we touched on earlier, probably similar to cooking, important in our early evolution for sort of like nutrition and food safety, and are really good incidentally at like extending the shelf life of foods. So like if you like take some milk and leave it out without it fermenting, it will like go off in a day. Whereas if you lacto ferment it into like a kefir or a yogurt, or then like press it into a cheese, it can last for like weeks or months or even longer. So it's because organic acids are actually like toxic to most microbe species.
You're kind of creating a natural preservative within the product.
You are literally creating a natural preservative. Yeah.
What are some other examples then of lactic fermentations that we're all familiar with?
So beyond the dairy ones, which is what like they're named after, sauerkraut, kimchi, basically any kind of like vegetable pickle is going to have a lot of lactic fermentation going on. There's a lot of important lactic acid bacteria involved in like sourdough and other sort of wild yeast and bread fermentations.
I mean, the name suggests you need lactose for this to happen. Is that true?
So no, they were I believe, initially discovered in like dairy products.
Okay.
So they're like, oh, you know, lacto is the like prefix for milk. So it's like, oh, well, the sugar we find in milk, we'll call that lactose, and the acid we find in milk, we'll call that lactic acid, and the bacteria that make that, we'll call it lactic acid bacteria. But then it turns out that they can use like lots and lots of different sugars.
Got you.
Even sometimes, like in wine, there are lactic acid bacteria that have the ability to eat like malic acid and citric acid and then make that into lactic acid. So like malolactic fermentation.
Yes.
Yeah, doesn't even involve sugar.
Yeah, okay, got you. And what are some of the molecules that are being created by fermentation? Because it can turn something very normal into something remarkably flavorful, can't it?
Yeah, so acids are the big one, but you'll also, you'll create a lot of like alcohols. So ethanol is the alcohol that we know the best, but there's also like, I guess you'd call it fusel oil in like a liquor context, but you know, isobutanol and some of these other ones that have these kind of like winey, pickly flavors. You'll also make organic acids, so that's where some of the like cheesy and funky notes come in, like butyric acid, hexanoic acid, getting into the goaty range of things. Then lactic acid bacteria will like combine those two groups of molecules together, the alcohols and the acids, to make esters. So you'll get like generally very interesting fruity qualities. Interesting because like alcohols and acids don't smell fruity at all, but when you put them together, you get these like apple and pear and other strawberry.
And those esters are the same things that are actually present, same molecules that are present within apples and pears, but they've been created through fermentation.
Yeah. So I guess they're like biochemically relatively straightforward to make. So some plants make them for fruit, and some bacteria and yeast will make them from other components too. And then you'll get some other things like diacetyl, which is really buttery. That's where actually like the buttery flavor of butter comes from. It's actually from cultured butter, like butter that you haven't pre-cultured doesn't taste that buttery. And then depending on what you're fermenting, what sort of material the microbes have to work with, you'll often see like in like wine and other yeast fermentations, a lot of the like fruity flavor won't actually develop until the fermentation, because fruits, actually lots of plants, will they'll like make a bunch of extra smell molecules, but then to like store them so they don't evaporate away, they'll like bind them to sugars. So you have this like pool of potential flavors that don't smell like anything. And then microbes have, they're really good at chewing up sugar, so they'll just like slice the sugars off of these smell molecules, and you'll get this like bloom of scent from it.
That's amazing. So in terms of the particular yeast or bacteria that's being used to ferment, you hear about brewers, winemakers, distillers sometimes using certain strains. So, and those strains are either selected or bred to accentuate certain characteristics in the fermentation. Is that right?
Yeah. So like commercial yeasts or yeasts that or bacteria that use like single strains. Either people will just have selected over time for like flavors that they like. There are also now some interesting strains available where people are like going in and specifically selecting for like weird and interesting flavor production.
And is anyone like genetically coding yeast? Is that a thing yet?
I believe there are some thiolizing yeasts. So these are like yeasts that will make sulfur compounds. Like some of that can happen naturally, but I believe there are some that have been like nudged genetically to do it available.
Because the flavor industry gets a lot of its aromas and flavors from fermentation as well, right?
Yeah, so this is actually one of the like earliest commercial uses of synthetic biology is taking, a set of genes or a pathway that makes that the vanilla plant uses to make vanillin, and then like inserting that into a yeast genome so that the yeast can like eat up sugar and then through its fermentation make a lot of like vanilla aroma, vanillin, and then you just like basically filter that out of the fermentation broth and sell it.
Oh, wow. So you're scientific advisor to NOMA.
Yes.
And you were involved in setting up the fermentation lab there.
Yeah.
So can you give me some like practical, not practical tips, imagine. No, some examples of some of the stuff you're working on there in fermentation.
Yeah, I mean, a lot of the early work on fermentation at NOMA was like figuring out, if we want to do a fermentation, but we don't have all the ingredients that traditionally go into it. So like if we want to do something like a miso, but we don't have rice and we don't have soybeans because we have, it's in our name, made this extremely limiting decision early on to like only use Nordic ingredients. So Nordic means like Nordic smell, Nordic food. Being able to kind of like look at a fermentation like miso where, you have a like very specific mold that you grow on rice to produce specific enzymes to then break down components in the soybeans. If you can, teach yourself how to use like Google Scholar a bit and like look through some of the research on this, you can, develop an eye for like, okay, well, it doesn't actually have to be a soybean if you have like a protein and a like proper protein source or proper like carbohydrate source. So that was like the original impetus for things like the yellow pea miso, which was like kind of the first fermentation in that vein that NOMA did, where it's like, okay, we don't have soybeans, we can at least look through like the Danish agriculture database for like what's the highest protein legume currently being cultivated.
Something similar to soybeans.
Yeah. And from there, once you've kind of got a category of ingredients in mind, it's kind of just a process of trying them out and seeing like how do you need to tweak the process a little bit to make it like work best for those things.
So you mentioned mold there,
Oh yes.
which is not a single cellular organism, right?
I guess technically mold is
it a fungi? I thought it was a fungi.
it's a fungi. I guess they make like, they're single cells, but they make like multicellular communities.
Right, okay.
So a mold is a filamentous fungi.
Right.
A yeast is also a fungi, but those are just like single cells, not interacting with each other that much. With a mold, you actually have cells growing into like a root-like network.
Yes.
Yeah.
And is there a distinction there between what it actually does in comparison to yeast and bacteria, the mold?
So whereas like yeasts tend to grow like on the surfaces of things or like in kind of like, fruits that have split open, molds are quite good at like digging into
they're buried.
stuff. Yeah, they're like burrowing things. So they'll sort of correspondingly, they have a lot of like specialist enzymes for like digesting as they burrow. So, yeast as fungi are really good at like taking sugars, turning that into energy. Molds will also do that, but they'll like have specialist enzymes that will like break down a starch molecule to like liberate sugar so that it can eat the sugar. So it has these sort of special, you can almost think of it as like a culinary ability to take things that it cannot eat and then transform them into things that it can eat. So break down proteins, break down starches, break down fats sometimes too. So it's like a kind of more advanced cellular machinery.
Yeah. Wow. I never thought of molds as being burrowing before, but then I guess it makes sense when you look at cheese and you cut it and it's got those veins going through it.
Well, yeah, the only part we usually see are the fruiting bodies of the mold.
Yeah.
But that's like, I guess.
Like mushrooms, right? You got the mycelium underneath.
Exactly. But only thinking of that as a whole organism would be like looking at an apple and then like not seeing the tree.
Ignoring the tree.
Also,
Yeah, sure. So during your time spent at NOMA and working with those guys, what sort of boundaries were crossed? What sort of new innovations did you witness or were you part of in food and drink?
I think one of the big ones, like a boundary that was fun to discard, was like the boundary between like a waste product and an ingredient. So, you know, cooking generally, but especially fine dining, tends to produce a lot of waste because you're like very selective about what, you know, what ingredients and what part of each ingredient you're serving to customers. And so like you end up with all this like offcuts and things like that. And, you know, at the same time, the sort of issue of food waste globally has been on people's minds for, you know, at least a decade now, kind of at the forefront. So like kind of being able to, so yeah, I think like the most important thing to come out of it is less like one particular innovation and more this like ability to like shift a mindset, shift how you are able to look at things. I mean, I think as cooks, you know, as culinary professionals, we tend to be taught just to like follow rules. Like you were there to perform a task and what someone else has figured out, and so you just look at it the way you've been told to look at it. So it's kind of counter to that to be like, well, let's like really think about why are we doing this and what are we doing, and should we actually consider a thing to be only this one thing, or could we look at it a different way? So the ability to kind of like shift a perspective from like, well, this is a problem we have to deal with, to like, well, you know, if we are using the best squid on Earth, then like the squid guts that we get out of it are also the best squid guts on Earth.
Yeah.
And so like, is that garbage, or is it like a new ingredient that we can figure out something to do with? And so that ultimately became like a squid garum, like a fish sauce essentially.
Okay.
Yeah.
And then was that fermented?
Yeah, well, so that's
If in doubt, ferment it.
Exactly. Either if it's got protein and starch, put it with koji and salt. If it's got sugar in it, try lacto-fermenting.
We can ferment this. That should have been the name of the NOMA cookbook, really. We can
maybe the next one.
Yeah, maybe. Because there is the fermentation book.
There
by,
is.
Is it David?
Yeah.
Yeah.
Dave Zilber.
Yeah, David Zilber. Yeah. Which you're involved in as well, I assume.
Yeah. No. I mean, so after Lars Williams and I like built the original fermentation lab out of like old shipping containers and garbage, you know, it was sort of this problem that like there were, you know, two people in the entire restaurant that knew how to ferment everything, and like no one else did. And so, you know, David, who I think at the time was working the grill section, you know, he came in and he had like physics equations tattooed on his arm. And he was sort of a natural choice for like, hey, do you want to do some weird stuff? Yeah, and clearly that worked out well.
Yeah. So, but he left, didn't he? He quit cooking, has he not?
Yeah, I mean, he's writing a book about microbes.
Right.
And
so he's gone deep. He's gone deep.
He's gone deep. He's really into it. It's going to be great. And he also, I believe he works at Christian Hansen, so which is one of the companies that makes cultures that people use. So he's still working in food, but not in a restaurant.
I've only been to NOMA once, and he toured us around the fermentation area when we went there.
Did you go to old NOMA or new NOMA?
Old.
Oh, okay,
Yeah.
So the
Yeah, it was cool. So that was the shipping container. It was definitely like garden sheds kind of situation.
Oh yeah. Well, so like there for a long time they're behind. Okay, so if you go to Copenhagen now, the area where NOMA used to be is like incredibly busy and touristy.
Oh right.
That is because there is a bridge that they built between Newhound, which is the like sort of harbor touristy area downtown, and that bit of Christianshavn. But the bridge was like, there was like an engineering error. So like all the bridges have to be drawbridges because Copenhagen's like an active port. So like ships have to be able to go through. And someone made a calculation error. So like the two ends of the bridge didn't meet. And it took like, I don't know, 8 years or something to fix. So for a long time, there was just like a construction site behind NOMA. So I think the thinking was like, well, if we just put a couple of extra shipping containers there, like no one's going to notice. And that became, you know, rather than doing koji fermentation in like 5 broken chest freezers in an attic. We got to do it in a slightly more controlled situation there.
Since we're on the topic of NOMA, René Redzepi, who obviously is the founder and head chef, he wrote the foreword to your book.
He did, yeah.
So tell me about the book.
Well, so I obviously have a background in science.
Oh, do you?
Well, no. I mean, like a formal background. Yes. Because people have all kinds of backgrounds. When I graduated college, I went straight into grad school and like learned all about flavor chemistry. And then, but I did that because I wanted to be doing, creative and innovative things with chefs and gastronomy. So after that, I was at NOMA for a number of years, and in addition to like experimenting with fermentation, kind of generally trying to figure out like, okay, there is like all of this information, this like ocean of information in the sciences on how to, like how various things work, how to do different things, like what of it is useful? Yeah. Because it is like, it can be very useful, but like you have to find the right piece of information, like explain it the right way. So I found myself like, one, figuring out what these pertinent kind of ideas from science were, and then 2, finding ways to communicate them, because like, there are some people who are chefs that have like master's degrees. Many people have not gone to college because you don't need to if you're going straight into like a culinary career necessarily. I mean, so, sometimes you'd be talking to people who like left school at like as a teenager to become chefs. And so like you cannot speak to them the way that you would speak to another scientist, which is kind of obvious, but like, anyway, so you, and by you, I mean me, you find yourself, trying to find like the right metaphors for like explaining things that are going to be really useful to people, and then explaining them over and over again to multiple people, and being like, oh man, I wish there was like a book I could give you. Like I have this stack of academic papers, but no one's going to read that, and there's too many. And then eventually you stop being stupid, and you're like, oh, okay, I guess I have to write that book.
Yes. So
between that and then for a number of years, I worked on a TV show on the Food Network called Good Eats, which I was a huge fan of in its initial run, a huge influence on me. So when Alton Brown reached out to be like, hey, do you want to work together? It was like an absolute, dream scenario. But in a situation like that, you're also explaining science to non-scientists. And in that case, you have 22 minutes to tell them everything they're going to know. Everything they're going to know about like some particular area of cooking and science. So you have to be like very, very selective about what you're telling someone and how you tell them that. So both of those things helped cure me of the curse of being an academic author, which is where you just try to throw in as much information as possible to give context, and figure out, okay, like how do I help people learn something useful?
Distilling it down and making it relatable is the trick, isn't it?
It is the trick. Yeah.
Yeah, the trick.
So so eventually the book kind of coalesced into there is all this stuff that we know about flavor in the sciences, and everybody, non-scientists, everybody loves flavor, but nobody knows how it works. Yeah. So what can we know about flavor that helps us be more creative?
And it's called Flavourama.
It is. Flavourama, a guide to unlocking the art and science of flavor. Mostly because all the sensible names for books about flavor were already taken.
That's a great name. I think it's really cool. It sort of suggests the kind of fun and exciting component of flavor.
Well, I feel like a lot of popular science books kind of like to tell people what to do, or they like take this position of like, well, I'm going to tell you everything that's right, and you have to, so I'm just like the correct way to do things. And like, I just don't think that's interesting or useful, and so I was hoping people would get excited about all the like crazy shit that you can do with the science of flavor.
And it's like you said earlier, none of it means anything until someone actually puts it in their mouth. And there's the whole psychology side of flavor, which we won't go into today, but that's...
Oh, no? No, sorry.
Well, I had Charles Spence, Professor Charles Spence.
Oh, yes.
Yeah, so he was...
iconic.
He was on the show. a little while ago.
Amazing.
So we did a lot of stuff on flavor
He would be the person to ask.
Yeah.
Psychology of flavor.
Yeah. Iconic indeed. Before we finish up, we touched on it earlier, and I just remember we need to go back to it. Are there any things that you've discovered in your journey through chemistry and biology and flavor that has changed the way you think about a food stuff, like for the better or for the worse, perhaps?
I think so. I was formerly trained as a chemist and a sensory scientist. So like knew about molecules and how to measure them, and knew about how to like get people to generate good tasting data. It wasn't until a bit later that I basically had to teach myself. But I guess this is what happens if you build a scientific career, like you branch out into other areas. Learning that these molecules that I was measuring, that I was interested in, that came from, like plants and fermentation, were so like intimately tied to our evolution, to the evolution of those organisms, to like their relationship with each other and with ecology. That really made me think about how, we like flavor as a hedonistic experience, but it has these like incredibly direct ties to ecology, to organisms interacting with each other, to place, to land, to like our turning into humans in the first place, that gave it this gravity that is really profound and interesting to me.
I agree. And it's just, I find it remarkable that until you have conversations like this, most people don't tend to give it a second thought. And a lot of people go their entire lives without really considering what flavor is and why they like it. They'll enjoy flavor. I'm sure they'll enjoy the food they eat. But to sort of take it back to the evolutionary journey that we've been on alongside these organisms and plants, and it's remarkable that we've sort of ended up in this place now where we can put something in our mouth and experience all these different emotions and sensations, and then put something else in and it's completely different.
Yeah.
And we can do it every day.
Mind-boggling, multiple times a day. I mean, we're also in a situation where we're like, oh no, plants don't have any flavor anymore, and also agriculture is taking the nutrients out of everything, and also agriculture is like bad for the planet. But how do we like reconcile all these things together? Well, like all of them are directly related to flavor.
Yeah.
Like they're not unrelated.
Yeah.
Talking about where flavor molecules come from. There's others that make like the really like sexy smells, and like quinces and roses and apples and like tomatoes, called myrisopenoids, and like they come from the breakdown products. I'm sorry, no, I'm getting like very technical now. Plants make them by breaking down large molecules called carotenoids, and some carotenoids our bodies can make into vitamin A. So like it is an essential nutrient, and the smell molecules that come from vitamin A carotenoids smell like more interesting and like 285,000 times more intense than the smell molecules made from like non-vitamin A carotenoids. Whoa. So if we're talking about like, the flavor of an apple is just to convince school kids to eat it, like, well, it is also a signal for whether you're getting this essential vitamin or not.
It's linked, like pretty powerfully linked. Wow.
Yeah.
Have you got any favorite molecules, like whether it be from an aroma point of view or for its veracity? Or, I don't know.
Well, so I do really like these myrisopenoids. Beta-Damascenone is one of my favorite ones. It smells sort of like if a caramelized apple got like distilled in a fancy tobacco shop. It's really good.
That sounds just quite tobacco-y. Yeah. As caramelized apple almost makes me think of tobacco.
Well, it's in tobacco as well, and like roses.
Well, where do we find this particular molecule?
You don't often find just that molecule, but it's an important kind of like contributor, but like it makes like good apples taste good. So like apples, wine, it's in like lots of wine, especially red wine, tomatoes. So basically any of these like sort of like fruits with a lot of depth of flavor, that often comes from myrisopenoids. There's another molecule that I'm a big fan of, called 2-acetylpyrroline. And this is one of those molecules that's both like, it can be produced by plants. It can also be produced by the Maillard reaction. So it's in, it's like the smell of popcorn. It's the smell of tortillas. It's also produced by basmati rice and pandan. So it's like that kind of like malty, slightly creamy, grainy aroma. But what's really like fascinating and like fucked up about it is that it's also produced by lactic acid bacteria and like certain strains. And when they sort of contaminate and ferment it in wine, it doesn't smell like tortilla or pandan. It smells mousy. So it's like the same molecule. So it's not even like, oh, like
Mousy.
Yeah, it's emphasizing something
What,
different.
like a rodent?
It smells like smelling a mouse. And it's like, I guess, both like just perverse to me, and that is amusing.
Yeah.
But also like, just as an example of like, no, flavor really is much weirder than you think. That's a good reminder.
One of my favorites is, I think it's isoamyl acetate, which is the banana.
That's the banana one. Yeah.
Yeah, and I have a feeling that bees chuck it out as a pheromone.
I would not be surprised if it did.
I think it's like a fight or flight response. They chuck it out, so you can't smell it in the air.
Oh, they don't produce enough of it?
They don't produce enough, but enough for a bee to be able to smell it.
Yeah.
You know?
Yeah. Amazing.
Yeah.
No, like insects do so much interesting communication through chemistry, things like that. But then one of the like first pieces of work I did with the guys at NOMA was when they were like they were like really interested in insects as a culinary ingredient. And this was back like the first summer that I talked my way in. And my now very good friend Lars was reading a paper about like entomology, about like insect signaling. So it was all these chemicals, and I was just sort of like looking over his shoulder, and I was like, "Oh, why are you reading a paper about like lemongrass and herbs?" And he's like, "I'm not. I'm reading a paper about ants." And I'm like, "Well, that is citral. That is the molecule in lemongrass." So yeah, It turns out that
because some like ants can be quite kind of citrusy,
and it's because they make the same molecule,
right?
Which is fascinating.
Yeah.
Yeah.
And especially if you've got a seasonal restaurant in Scandinavia.
Or you're trying to, how do we find ginger and citrus flavors? Yeah.
Yeah, wow.
Ants. Yeah.
Oh, that's cool. So what's next? What are you working on at the moment?
Well, my book came out in March, so I've been doing a lot of promo for that, which has been amazing. It took. I mean, you've written books, you know that it like lives in your head for a really long time, and then all of a sudden, all these people are talking about it, which is fantastic.
Yeah.
And I've been. I'm at the point. Well, I don't have children, but people often talk about it similarly as like you have one, and you're like, I'm never going through that again, and then eventually you get to the point where you're like, oh, let's do another one. So maybe some more stuff in that vein. I'm also doing some like fun consulting projects. Some in like the liquor space, some communicating about flavor with some larger brands, which I can't talk about yet. But getting my foot in a lot of.
Are you NDA'd up on the liquor bit, or can you say anything about that?
Oh, I am. I'm sorry.
Okay, no worries. That's all right. Maybe when whatever it is comes to fruition, you can come back on and tell us about that.
Yeah.
Cool. Arielle Johnson, amazing. Thank you so much.
Thank you. This was like a super fun conversation.
Good. I'm glad you enjoyed it. Where can we find you? Where?
If you go
Flavourama, and then
Flavourama, coming out in Germany, actually at the end of November, in German. Not in English.
Oh wow!
Yeah,
straight in a translation.
Yeah,
Brilliant.
exactly.
Yeah.
If you ariellejohnson.com and Instagram at Arielle_Johnson is where I'm most active.
Thank you so much. That was fascinating.
Thank you.